Hello everyone, welcome back to my videos. This video is on glycolysis, an aerobic reaction, presented by me, Dhara Fatnani. Glycolysis. Glycolysis is a part of metabolism. Metabolism includes catabolism and anabolism. Anabolism is majorly seen in plants. Example is it uptakes CO2 from atmosphere, water from soil and sunlight from sun, converts it into glucose molecule. Example of catabolism is seen in animals. Animals take glucose and convert it into pyruvate and ATP as an energy source. The name glycolysis means lysis, that means breakdown of glucose. Enzymes. What are enzymes? If we take two substrates, that is A and B, we make them react with each other. It is not compulsory that we get a product. See, A and B substrates are still individually substrates. Whereas if we make these substrates A and B react in presence of enzyme, then it is converted into a product. Here the enzyme used would be specific for substrates, and the product formed has different characteristics as compared to the substrates. Enzymes. Which kind of enzymes which we need to see in glycolysis? So the very first kind of enzyme that we need to learn is enzyme with kinase activity. That means an enzyme is capable to transfer a phosphate group from one molecule to other molecule. For an example, we see ATP, adenosine triphosphate. This is in presence of kinase enzyme, reacts with it and leaves a phosphate group and is converted into ATP. This phosphate group would then be transferred to another molecule. This is the function of kinase enzyme. Other kind of enzyme that we are going to see is enzymes with dehydrogenase activity. Dehydrogenase, by its name, it means that it removes a hydrogen atom from the molecule or from the substrate. For an example, if we have a substrate and we make it react with dehydrogenase enzyme, then this substrate would remove a hydrogen atom and it would transfer this hydrogen atom to NAD+, and this NAD+ would be converted into NADH. The hydrogen atom here is taken from substrate. This was the activity of dehydrogenase enzyme. Moving forward, let's see the fate of glucose. The first fate of glucose is pyruvate. This is by the oxidation process and the pathway is known as glycolysis. This pathway we are going to see. The other way that a glucose could be used is its storage. Glucose is stored in form of glycogen, starch and sugar. Glycogen in animals in liver. Starch could be seen in fruits or in vegetables of plants. Sucrose is also seen in plants in sugarcane. The third path of glucose is also oxidation, and this pathway is known as pentose phosphate pathway. In this, glucose is converted into 5-carbon structure ribose 5-phosphate. Glycolysis occurs in cytoplasm of cell. That each and every pathway of biochemistry has its specific location, and glycolysis here is majorly seen in cytoplasm of the cell.
This process is continuous and it does not depend whether you are eating or you are not eating. It is continuous as your cell needs energy all time.
Moving now to glycolysis. The substrate of glycolysis is glucose. We have an enzyme hexokinase and it has kinase activity. So it converts an ATP into a ADP, transferring its phosphate group to glucose molecule, and glucose is converted into glucose 6-phosphate. 6 is because the phosphate group is attached to 6th carbon of glucose. Hence it is known as glucose 6-phosphate. Now, this glucose 6-phosphate in presence of isomerase enzyme is converted into fructose 6-phosphate. Glucose was an aldehyde, fructose is a ketone. Now, fructose 6-phosphate in presence of phosphofructokinase enzyme would be converted into another molecule. But let's see what is the function of phosphofructokinase. Phosphofructokinase converts ATP into ADP. Again, a phosphate group is taken from ATP, and of course it would be transferred to fructose 6-phosphate to convert it into fructose 1,6 bis-phosphate. Bis is because now there are two groups of phosphate. First group is attached to the first carbon of fructose, and the second phosphate group is attached to the 6th carbon of fructose. Hence it is named as fructose 1,6 bis-phosphate. Fructose 1,6 bis-phosphate in presence of enzyme aldolases is converted into 3-phosphoglyceraldehyde and dihydroxyacetone. Here, by the enzyme aldolases, fructose, that is 6 carbon molecule structure, is broke down in 2. So, we get a 3 carbon molecule structure, glyceraldehyde or dihydroxyacetone. Dihydroxyacetone is easily convertible to glyceraldehyde. Hence, here we are going to see 2 molecules of glyceraldehyde in the further reactions. Of course, fructose also had phosphate groups with it. It gave it phosphate group to glyceraldehyde. 2 molecules of glyceraldehyde each got 1 phosphate group. So it is named as 3-phosphoglyceraldehyde. We can see at its structure. The phosphate group is attached to the 3rd carbon atom. Moving forward, 3-phosphoglyceraldehyde in presence of 3-phosphoglyceraldehyde dehydrogenase removes a hydrogen atom from the aldehyde and donates it to NAD+. So NAD+ is converted into NADH. Here you can see that the aldehyde CHO is converted into a carboxylic acid, which is named as 1,3-bis-phosphoglycerate. Aldehyde group is converted into acid, that means glycerate. We can also see that a phosphate group is added to glycerate. That means phosphate group is not added by a kinase enzyme, it is added from a free phosphate that was present in the cytoplasm. Moving forward, 1,3-bisphosphoglycerate in presence of another kinase enzyme converts ADP, that means adenosine diphosphate, into ADP. Here, this kinase enzyme is not taking phosphate group from ADP, it is donating phosphate group to ADP and it is synthesizing a ATP molecule. From where the phosphate group is coming? The phosphate group has came from 1,3-bisphosphoglycerate, and this 1,3-bisphosphoglycerate is now converted into 3-phosphoglycerate. Also, you need to see here that 2 molecules of ATP are formed because 2 molecules of 1,3-bisphosphoglycerate are donating its phosphate group. In this reaction, The phosphate group is taken from the substrate. Hence, this is known as substrate level phosphorylation. This is the first substrate level phosphorylation occurring in glycolysis. Moving forward, 3-phosphoglycerate is converted into 2-phosphoglycerate by the enzyme isomerase. Isomerase changes the configuration of few molecules. Here, the phosphate group that was attached on the third carbon of glycerate has moved to the second carbon of glycerate. Now, this 2-phosphoglycerate in presence of enolase enzyme is adding a enol group and is converted into phosphoenol pyruvate. Phosphoenol pyruvate in presence of kinase enzyme converts a ADP into ATP. Again, a phosphate group is donated by substrate. So this process is also known as substrate level phosphorylation. As you can see, it is second substrate level phosphorylation. Two ATPs, here also two ATPs are formed because two molecules of phosphoenol pyruvate are donating its phosphate group. Moving forward, let me ask you this question. How many ATPs are formed from one molecule of glucose only in glycolysis pathway? And I'm not asking a whole or that by breakdown of glucose how many ATPs are formed. That is a different question. But in glycolysis, how many ATPs were formed? So, what the answer would be? Let's see few pathways. In glucose, the very first reaction, we consumed one ATP molecule so that we can add a phosphate group to the glucose. Also, there was another reaction where we again added a phosphate group to fructose 6-phosphate. Okay, see, this is a glucose molecule, and we added a phosphate group to it on the 6th carbon. We took this phosphate group from ATP, we consumed ATP. In the second reaction, fructose 6-phosphate is also converted to fructose 1,6-phosphate bis-phosphate. We again consumed an ATP. So 2 ATPs are consumed in this process. Okay. Now moving forward, in the first substrate level phosphorylation, we synthesized 2 ATP by transferring phosphate group from the substrate itself. So 2 ATPs were synthesized here. 2 ATPs were consumed earlier, so it is neutralized. Till now, no ATP is generated. Moving forward to phosphoenol pyruvate reaction, this was the second substrate level reaction, and here also we generated 2 ATPs. So in total, if we see, we got 2 ATPs. But there is another reaction. 3-phosphoglyceraldehyde was converted into 1,3-bis-phosphoglycerate. Here the main thing is NAD plus was converted into NADH. So this NADH will go to electron transport system and would generate 2.5 ATP each. As there are 2 NADH, of course, total number of 5 ATPs would be generated in future. So, one molecule of glucose yields 2 ATPs plus 5 ATPs from NADH, and it is in total 7 ATPs in glycolysis pathway. Also, you can say it in a different way that only 2 ATPs are formed in glycolysis, as NADH is not a direct way of ATP until and unless it goes to electron transport system. Second is 2 substrate level phosphorylation takes place in glycolysis. Okay, this is an important point which could be asked in various competitive exams. Moving forward, thank you for watching. If you have any doubts or if you think that I have left something to explain, you can surely ask me in the comments. Thank you. Don't forget to like, share and subscribe. Also recommend this to others.